Thank you for joining us today. This is the last regular botany seminar of spring 2022, and we're very happy to close this cycle with Professor Chris Gilligan from the University of Cambridge. I also wanted to thank all the organizers for making this possible. As a courtesy for participants, let me remind you that questions should be posted on the chat, and you could also raise your hand by selecting the participants tab, and we will unmute you. As you may know, plant pathogens and diseases continue to challenge crop protection efforts at the local, regional, and global scales, but fortunately we have plant disease epidemiologists. And Professor Gilligan is a world-renowned authority in plant disease epidemiology. He has made highly influential contributions to the theoretical framework for plant disease invasions, persistence, and variability, and not only for plant pathogenic agents, but also he has recently been working with animal and human diseases. He has received numerous honors. He has, for example, been named Fellow of the Royal Statistical Society, President of the British Society for Plant Pathology, Director of Research at the University of Cambridge, where he continues to lead research programs in epidemiological modeling. He was recently appointed by Her Majesty the Queen as a representative of the Board of Trustees of the Royal Botanic Gardens, and has served on numerous UK government committees, and is in demand for national and international service. Without further ado, please join me in welcoming Professor Gilligan. Chris, it's a pleasure to have you here.
Thanks very much, and thanks for the opportunity to present and also to chat to many of the team this afternoon. I'm just sad that I was unable to visit you in person, but maybe sometime in the not too distant future. I'm going to talk about models for emerging and recurrent epidemics, and I'll pick some of the recent work that we have been doing. There's a team of modelers here in Cambridge, and some of our collaborators from a little bit further away, Stanford, through to the Met Office. Some have moved now into industry, but we're still collaborating. And I'll pick out the particular people who've made the special contributions as we go through. I want to think about the challenge and particularly about extending the practical applications of the sorts of models that we work with. We've developed a toolbox and an epidemiological framework that encompasses, I think we've applied it to about 20 different host pathogen systems, but more recently I've become very focused on what's happening in sub-Saharan Africa and South Asia. We can look at a range of problems here with cassava, for example, cassava mosaic, cassava brown streak, and the whitefly vector. We have problems with the whitefly of superabundance, increasing impact, extending range. Moving out, we can see fall armyworm, which we're not working on at present, but maize lethal necrosis, where we're developing some outline models, a new problem. An invading problem, very rapid. Panama race TR4 and Panama disease has been introduced for the first time into sub-Saharan Africa in Mozambique. Very threatening for the spread from there. Aflatoxin on maize working in India. More problems coming through, which we'll talk about: wheat stem rust, wheat stripe rust, and of course wheat blast. All of these we're developing models for. Banana bunchy top has prevented growth of banana crops in large parts of Africa, and though we haven't started work on cocoa swollen shoot, we've been approached a number of times to look at this. Also desert locust. Over the past 18 months we have been working on there's been a big upsurge of desert locust in East Africa, and working on developing some models for that, and that is one of the hardest systems that I think I've ever worked on. I'll focus today on these, however, cassava and the wheat rust, to illustrate some of the work that we're doing. And let's think about what sort of approach we take. So suppose you have an invading pathogen and you want to assess the risk. Where and how fast? What's the impact on livelihood and health? What are the options for management? And how should we prepare for this? And imagine that you're adopting an aerial view and you're looking from above, and we're thinking about the potential for spread, the impact and the options for control. And this is part of Zambia here, but we're also thinking the same applies whether we're in a developing region or a developed region, how this would scale up. So you think about developing a strategy for a region, a village, and then a region like this, and scaling up into an entire country. What do you need to do, and how should you stratify that as you go from households, villages, districts, to national and regional scales? So we really need to think about a simple way in which we can deal with virtually any epidemic and indeed pest infestation that we encounter. And the way we approach it here is by having a basic form of mechanistic model. Take a susceptible, which could be a plant, but it could also be a field. And so it depends what system you, what scale you want to work at. It could be larger than a field. That's our unit, and that moves from susceptible. It becomes exposed during this latent class, so it is not infectious at this stage. Then it becomes infectious, but first of all, very often cryptically or asymptomatically infectious, then symptomatic, so you can detect it, which may be visually or it may be by molecular or remote sensing, we hope, in the discussions today. And then the plant or the field becomes removed because it's harvested or it dies. We have secondary infection as the principal spread here, where the epidemic is fueling itself, but with some primary infection coming in from outside the system. So secondary infection within whatever our system is, something kicking it from outside. So even if you control what's happening in here, if you don't manage what happens from outside, you'll have problems. We have a latent period and an incubation period, and a cryptic period, infectious period. These are the common sorts of characteristics that you would have with human and livestock disease, as well as with crop disease. We now need to think about introducing uncertainty and allowing for space. So let's think about how we might do this. First of all, let's think about space and spore dispersal kernels. Here we can see spore clouds emanating from Ethiopia, Yemen, and Iran, very different characteristics, and I'll explain those in due course. We may have a consideration too, where you're just thinking about distance and you're thinking about the rate of infection, and you have a disease dispersal kernel. That is, you're thinking about how far can infection spread from a particular site, characterized by dispersal kernel rather than by spore dispersal kernels. This is dispersal kernel for the affected disease. You might have spread networks, so that there can be trade networks, for example, that are spreading whatever you're concerned about. And we often deal with metapopulations, so you have an epidemic developing not just in one field, but in regions of fields, and then spreading from region to region. What about that question about uncertainty or stochasticity? Here's a simple illustration. We've got time running on here, and we've got the number of infecteds, which can be infected plants or infected fields or infected villages or whatever it might be. Here is a deterministic model which describes that infection and the epidemic. It's increasing, then it decreases and would eventually die out. That is deterministic. So you write the equations down and you'll always get the same answer. But life and death are not quite like that. And what we illustrate here in the pale blue and also this region here is a stochastic model where we are looking at a range of outcomes. So we let the model run and we're allowing for the intrinsic uncertainties in the system. And so disease could be as bad as this or as good as this, where think about if you set up a whole lot of field plots and you inoculated them, you would not get the same epidemic in each of those field plots by any means. You would get a lot of variability, and this is capturing that variability. And importantly, some of those, quite a large proportion in this, because it was just, we all know what R0 is now because of COVID. We were just close, slightly above R0, the threshold for the epidemic taking off in this case. But a lot of the replicants did not take off. And this becomes important too in thinking about how you control disease. So you have to think about how bad can it be, how good can it be? Will in fact some epidemics not take off at all? And I mentioned COVID. I was talking to some individuals earlier on today and saying we never rush into print, but this is one of the fastest papers we've probably ever produced and had published. It came out very early. We started this work in April in 2020, where we were asked about masks. And I'll just illustrate one of the examples here. And the point about this is very similar model is used in human epidemiology as compared with plant epidemiology. We have time running along here in days. We have the number of cases in millions. We were thinking about the UK. We have total removed, which is an epidemiological term for the total number of people that would have been infected over time, and the total who are actively symptomatic, which is a little hard to see, but you can see the red epidemic was kicking along here. At the time, for the parameters that we were supplied, we thought that the epidemic would, if we introduced the lockdown periods, would actually drop away, but then you see it comes up again, and if I change this to a log scale, you would see that it would also come up repeatedly, but the focus here was on masks. Here is a region in which you have lockdown, no lockdown, and we predicted that it would be necessary to have a series of lockdowns, and still getting half the UK population infected in the absence of using face masks. If we do use face masks, we can see that the total actually is still quite high, 25%. We've affected what happens in the first lockdown region by comparing here and here, but still quite a high level of infection. We distinguish between the mask, that's the total. We distinguish between the non-mask wearers where the infection would be higher, and the mask wearers where the infection still would occur but would be lower. But as you go from no masks to 25, quarter of the population wearing masks some of the time, series of sequence of lockdowns, thinking about the UK, as you move above a certain threshold level for the benefit of wearing masks, you see 50% really made a big difference. Depends on how much then movement and spread there is between times, and 100% very significant. So this led the UK government, in part anyway, to introduce a requirement for mask wearing. A message is that, excuse me, because I've been talking all afternoon, I need to drink every now and again. It's only water, however, for sure. This is a case where you use a relatively simple model, you allow for uncertainties in the system, and it can be used to inform policy. So I'm now going to move to wheat rusts and think about what can we do with wheat rust. And we'll be considering 3 rusts: stem rust, stripe, and leaf rust. and thinking about continental spread, and then can we develop any real-time forecasting system? So let's take the continental spread, and this began with wheat stem rust. It has since been extended, as I'll talk about as we go through. Work with Marcel Meyer, who was a PhD student with me. We're continuing to work with Dr. Dave Hodson in CIMMIT, and also with colleagues in the UK Met Office. And full credit goes to Marcel for the work that he did originally in his thesis on this. Now let's think about sites at risk, and on this movie, which I hope you can see moving as you can, what are we looking at here is the sowing date and how there is a change in the availability, looking at what's happening in the region here from Africa up into Middle East, West Asia, South Asia over time. Let's run that again, and we see that's changing relatively slowly. See the months change here. But environmental conditions, which are looking at the suitability across the landscape for infection, are changing very rapidly on a within a 3-hourly or sometimes hourly basis. So slow change here, but it does mean that some regions are unavailable during part of the year, and then rapid change in the environmental conditions. So let's think about pathogen dispersal. And here we're using models that were devised originally for the spread of radionuclides and pollutants of various sorts. So you are looking at the turbulence of the wind. We introduce the environmental conditions for meteorological conditions, and then run what's called a Lagrangian dispersion model, which is tracking very many, often billions of spores in this case, or groups of spores, through time. So we have this time constant, and then what is happening where this is changing. You can see this changing daily here, monthly, on the hour, and we're thinking about the correspondence of these. We have dynamic land interactions then between a host landscape, between the environment landscape, between the pathogen dispersal. And one also needs to allow for the social landscape in what are growers doing in these different countries. So we looked at that over analyzed a 12-year period of historic data. And the data were changing at 3-hourly intervals, and at initially at 17-kilometer resolution, and then increasingly or decreasing it, depending how you look at it, at 10-kilometer resolution across this entire region here. And what do we see? First is that there are, so we're looking to see who could have exchanged spores with whom if they were infected. And we see 1, 2, 3, 4, 5 colors here. There are 5 tightly or well-connected regions. Take the Rift Valley region into East Africa here, that's one region. The Southern African region, here is another, but very little contact between there and here. Some contact between here, getting up through Saudi Arabia into the Middle East here, some into Afghanistan, and then South Asia here. So different groupings. The size of the circles indicate the net movement of spores. So we can see Nepal here has a lot of spores moving in and out, and the shading indicates, is it predominantly an exporter or an importer? So we can see from this there are 5 strongly connected regions, high degree of atmospheric spore transmission. Nepal is a major donor. Yemen, a very troubled country, unfortunately, is a critical donor in terms of acting as a stepping stone in and out of what happens here in the Rift Valley and into East Africa. So you think about UG99, new strain that was identified back in 98, 99, big worries about where that was going to go. We can see now, and we've done a lot more work since this, the major routes by which new strains come in, and this can help us to plan control strategies. Uganda, it turned out, was a spore receptor rather than a donor. That was a combination of the dispersal modeling, the meteorology, and some network analysis that we did. That tells us what was happening retrospectively in terms of historical data, but can we use this to predict risk? We're now going to think about a source country, and this is running right through from Bangladesh, Nepal, right through, as you can imagine, down through Middle East, East Africa to South Africa. And then we think about each of these in turn being a receptor country. So in looking at the historic data for how much spore movement there was between particular countries or regions, one can look at the daily mean proportion of airborne transmission during wheat cropping periods and allowing for suitable conditions. Take your mind back to there's no point just looking at the meteorology if there's no wheat in the ground, because you're not going to get a lot of spore reception or donation then. So here we have a connectivity matrix of who would exchange spores with whom in terms of countries when there is wheat in the ground. And this then can be used to say, supposing we're interested in Yemen, and what's the threat to Ethiopia? This is averaged over an entire growing season, and we see that the threat to Ethiopia is remarkably high if we have a new strain occurring in Yemen. That's Yemen being the source country, Ethiopia is the receptor country. There are various things that one can do with this to look at small outbreak at the source. We can distinguish then a moderate outbreak or a large outbreak, and you see that the amount of spores that you've got present can clearly affect the risk to many of the neighbors. I should also say that those 5 groupings that I indicated a couple of slides ago there's a lot of support for those two from records of different strains and races of the pathogen that have been recorded in the individual countries. So it is encouraging that what we've identified from the historic data are indeed showing up in terms of what's really found on the ground. Okay. So can we move to real-time forecasting? And we're working with CIMMYT, with the UK Met Office, the Ethiopian Institute of Agricultural Research, and ATA, the Agricultural Transformation Agency in Ethiopia. The point about ATA is they have messaging systems where they can actually access very large numbers of growers of the order of several million. Note too that Ethiopia is the major wheat growing country of sub-Saharan Africa, and this has been extended in this season to Kenya, interacting with CALRO and CIMMYT. A lot of people have been involved in this from Cambridge, from CIMMYT. Some of you may know Dave Hodson, tremendous scientist. Yusuf Alameu works in CIMMYT in Ethiopia. And 2 new postdocs have joined who have both got meteorological backgrounds but are now well versed in epidemiology, as we have as they join the team here in Cambridge. So what are we doing? We're thinking about surveillance data, which I'll explain in a moment. Being supplied, we know here's the Bali region, BALE region in Ethiopia, very often the source of early infection there, and then using meteorological data, we can predict where the pathogen is likely to go. So what have we got? We have numerical weather prediction from the UK Met Office, and that gives 3 hourly meteorological outputs on that 10 by 10 kilometer resolution over the next 7 days. Is 7 days long enough? Yes, it is, because if you can give growers a 7-day warning that rust is likely to occur in a region, they then have another 2 weeks, so that's a total of 3 weeks in which there is time to put fungicide on. So we have surveillance data that come in, meteorologically driven models that predict where the spores are going to go, and also meteorological data that we use in a model for infection efficiency, which is looking at the temperature and the environmental conditions that would be favorable for infection. Putting these together, which I'll come to shortly. And as I indicate, that gives a forecast of spore deposition, suitable conditions for infection. 1 to 6 days is now 1 to 7, but essentially or effectively that is 21 days. And a reminder then that Ethiopia is still the largest wheat producer, and of course with the very sad events in Ukraine, wheat is going to be a very precious commodity over the next season. Here's an example where we have spore deposition at a particular time predicted over the next 7 days. And let me just check, 5, 6, yes. We take the spore deposition, we look at the environmental suitability for infection, you see the scales up here, and then put those together so you get the integrated risk. So although the spores might be widely dispersed, the environmental conditions are not so widely dispersed, and then here are the regions that are major risk by integrating those two. Does it work? The overwhelming evidence is yes, that it does, and it has changed the behavior of growers in Ethiopia, with hundreds of thousands of growers coming on. We'll be moving with there and elsewhere, we hope, up into the millions shortly. And I'll show one example, not the best of graphics here, but there was a very severe outbreak with Digaloo race that developed in 2010. That was a huge epidemic year when there was no early EWS's early warning system, and we're looking at stripe rust in this case. The developed for stem rust, we now predict stripe rust, stem rust, and leaf rust. The red indicates, as you can see, high levels through to green low levels in that 2010 epidemic year, which was truly devastating for Ethiopia. Massive loss of yield, massive expenditure on fungicides that were bought and applied too late using foreign exchange that the country could ill afford. Didn't work. 2021 we considered was a potential epidemic year. New strains once again, PSTS 16 of stem rust identified from the Aarhus team and CIMMYT. Equally well, there'd been a new strain of rust, a race of rust I should say, coming in 2010. Same thing in 2021. Environmental conditions extremely favorable, but in fact very little, with some infection in some regions of course, but compare this with that. We're now analyzing a lot of data in terms of purchase of fungicide. Growers actually got fungicide on at the right time in the right place, and we believe that this has saved a major epidemic. So let me just take this a little bit further to real-time forecasting in South Asia, again with Jake and Tommy and Richard Statt, who's been involved in a lot of the research. Again, CIMIT with Dave Hodson, Tim Krupnik involved locally, and Will Thurston and Sarah Millington from the Met Office. And many involved from Bangladesh wheat and maize research. I must put a W, capital W for there, I see. The Department of Agriculture in DAE and the Nepal Agricultural Research Council. So work from Will Thurston at the UK Met Office, looking at the breast threat, stem rest in this case, but we're looking at stem, stripe and leaf here. This shows very nicely what is happening in terms of the deposition of spores in the left of those two movies. And then the particle height of the spores. We're in a highly topographical region, a lot of topography there. Sorry, the maths is topology. And thinking about where do the spores go? Are they going to survive? We assume that the spores, we've adapted the models to allow for UV radiation, actually killing the spores. They last for up to 3 days, possibly a bit longer in the dispersion. So we want to understand how these move, and we want also then to think about the topography and work with Marcel Meyer, who should appear in a moment on this, has taken the data that we get out from the Met Office for the historical and also for the predicted spread. And we can see that as we run this through time, and this 3D animation, let's just run it again. We can see that first there is the spore extends several kilometers firstly into the altitude, many hundreds of kilometers horizontally. The winds are directing the majority of the spores eastwards, but later into the Arabian Sea. Marcel Meyer, who moved from Cambridge to University of Hamburg, has been developing this, and we've also been adapting this to think about how desert locusts move, because as you think about desert locusts moving, they are not so much interested, they are affected by the wind turbulence. Some behavior happening there, but also as they get quite high, they will tend to try and move down, so that we need to allow for those high temperatures at height. What can we do then in predicting what's happening in South Asia? We can think about the stripe breast in this case. What happens when you have early levels of infection up here in Pakistan? What's the threat to Nepal and also to Bangladesh, which are the two countries that we were particularly focusing on initially, looking at historic data, but also contemporary data. We can then look at this in terms of what happens by looking back at the historic data. Where are the regions are particularly at risk if we're thinking about stripe rest and we're thinking about stem rest? Note that while these are both rusts and quite similar, in fact the behavior is not identical. And out of this we can identify first for stripe rest that we can consider different regions becoming, first of all, being the donors, and identifying the interregional dependence. So if we have an outbreak here, it's relatively restricted because of the environment, because of the meteorological conditions. Region 4, however, and region 6, when we're thinking about stripe rest, very high risks to surrounding areas. And again, with our focus particularly on Nepal and Bangladesh, but we hope to do the whole of South Asia. We've just brought in Pakistan. We hope soon to have Afghanistan data. you can see the different risks, and let's move between stem rust and stripe rust. So you see marked difference in terms of the risk with respect to stripe rust or stem rust to occur. And we do think that new strains of stem rust will be a major problem in these regions. So we have an early warning system that's been extended from Ethiopia into Kenya and also now into Nepal and Bangladesh. What's at the basis of this? Let's review it. There is surveillance, so near real-time data, and I know there's a lot of interest in Purdue in getting these near real-time data. This is by visual observation at present. I know you're particularly interested, as we are, and there's a part of this project which is looking at remote sensing, both from satellite and from drones. Crowdsourcing. So what do we do with lots of data coming in from farmers, where we have, particularly bringing in Bangladesh, upwards of 4 million subscribers. The WeTrust Toolbox, Global Data Management System at Aarhus. Data coming in to CIMIT, so we have a central early warning unit. It's the same system in Ethiopia. We then get a 7-day forecast from the Met Office in Cambridge models. So the survey data goes in, we update the models daily, and then we produce a 7-day forecast on each day. That enables the production, partly automated, not yet entirely automated, of weekly advisories, and then alerts that go out to the farmers and action. Actually, I've put in an Ethiopian farmer visiting Nepal and Bangladesh there. Let's, over the last section of the presentation, let's turn now to a different disease, cassava brown streak. It's a virus. There's been a major epidemic in East Africa. It's spread by trade and whitefly. And the question that we were originally asking is, what's the threat to West Africa? Rich Stutt has been involved in this right through. David Godding as well, a PhD student, now a postdoc. Rich also did his PhD with me and now has been a postdoc, an advanced postdoc for quite some time. And Anna Shinishevska, who has left recently to take a job in CABI. So these three particularly. What we want to think about is, can we develop a stochastic epidemiological model? And can we predict the spread to Central and West Africa? We're not alone in doing this. We've had a lot of collaboration in East Africa, notably Titus Alikai and his team at Naro in Uganda, also Patrick Chikoti in Zambia. And this was originally a cassava diagnostic project funded by the Gates Foundation, led by, Joseph and Dungaroo. And in West Africa, Justin Pitter leads the WAVE consortium, which began as the West Africa Virus Epidemiology, WAVE. It's subsequently brought in Central Africa, and originally with these 10 countries, well, extending to 10 countries, and within the next couple of weeks we'll be extending further to include 14 countries. So it's these that we're looking at. What's the risk to these, using some information from what's been happening in East Africa. It's really important to be able to work out where cassava is grown, and Anna Shinishevska produced a cassava map, because MapSpan is good, it's where we all start, but it wasn't sufficiently detailed. Anna's used a range of sources of data to produce, as best we can, a map for what happens in terms of the density of the cultivation of cassava across sub-Saharan Africa. There are also data on the abundance of the vector. Here you see whitefly, very high levels, in Uganda and Rwanda, some high levels in some regions of Nigeria here and interpolated elsewhere. We would like to have a lot more data on the availability of this vector. And just before I go to the model, so what are we going to do here? Essentially, we're using a susceptible infected type of model. So it's a classical epidemiological model with a dispersal kernel. And I remember I showed you those dispersal kernels early on. And using essentially those simplistic concepts from the epidemiological toolbox, setting an epidemiological framework, taking the model and then estimating parameters from a survey. And let's move to that now. What I'm going to show you in here are a series of data from 2005, because we were benefited from having historic data that we cleaned up with our Ugandan colleagues. And we have training data for a number of years that are used to parameterize the model. Here are what the data look like, and we'll run through these. There were surveys throughout Uganda. Red is positive, green is negative. We're simulating from the model here what the model would have said about what was happening here in terms of training the model. The gray areas are showing the density of cassava fields in these regions. So 2005, 2006, and note that as we go from 5 to 6, the surveys occur in different places, so we're having to allow for the fact that in 2006, though we knew something about 2005 here, we didn't know what was happening in 2006 from what we did know in those 2005. So 2006, 2007, 2008, 2009, 2010. You can see as we flick back through those, I hope that the simulated data and the survey data are doing really well. That's encouraging. But we've used those data to parameterize the model. So you would hope, though it doesn't always happen, that that would be possible. And for those only technically minded, we're using approximate Bayesian computation here to estimate the parameters. So we now go to test data. So remember that was up to 2010 using historic data. Let's go to the test data. And as we go through 2011, real data simulated from the model has not seen these data. And again, what's happening down here is a different epidemic and not one that we were attempting. It's a whole different characteristic on the coast here in Tanzania, not something we're trying to simulate. 14, 15, no data in 16 and 17. So this is encouraging. The test data actually illustrate quite well, not entirely, but quite well. In fact, very well, I would argue. What was happening in data that they haven't seen. So we fitted the data 2004 to 10, successfully predicts the results 2011 onwards, and we use the model to predict spread, to inform surveillance, and to compare control scenarios. I'll show you a couple of examples to finish off with here. Let's take risk maps and think about that westward spread towards wave countries. And as we run this through time, you see the epidemic developing here. Time is changing up here. We see the spread getting through Central African Republic down into DRC, and we very successfully, the model predicted when the disease would arrive in Zambia. You see once it gets, let's just run that again. So once it gets and begins to spread across here, we can see as we run through time, once it gets to here, very rapid spread because of the high density of the crop. That's if it were to spread across the continent in such a way. Of course, we also need to, let me just emphasize, it's a stochastic model. You know that it's parameterized, it's modulated by the host landscape and the vector density. We introduced a surveillance app, working very closely with our colleagues in Africa, now used in 17 countries. So we're getting real-time data. We could, of course, have direct introductions, say in a port, people bring the disease in, much as has happened with citrus greening in the States. And we can see in just 10 years, now we're running it out a little bit longer, potential for very rapid spread in the countries. Is there hope? We've run some scenarios where we talk about different surveillance approaches. We can think about low surveillance activity, partial resistant varieties and low uptake of those, high surveillance detection, partial resistant because the chance of full resistance is not high at present, and high uptake rates. Politicians are very worried when they see those previous maps in all of these countries, but when they see that potentially science can help, this is very encouraging. One further point, I've shown risk maps. So a risk map is essentially where is the disease most likely to occur given what we know now. A hazard map is how fast would the epidemic spread if it were introduced in a particular location. So we do this by initiating an epidemic here, running the epidemic over 5 years and see what happens. Then we initiate at the next point, running from there at the next point. Doing this, which is computationally intensive, as you can imagine, across the whole of sub-Saharan Africa, it is possible to generate a hazard map, which is essentially saying, and let's focus here, if the disease, the pathogen, were to be introduced in this region around Yaoundé, you can see high level of spread, a high hazard anywhere in Nigeria, lower level as we get out into the Ivory Coast, Cote d'Ivoire here, but back in Uganda, Rwanda, Burundi, and parts of Tanzania and parts of Kenya, again, high hazard. There's all the potential of whitefly, of connected hosts, of environmental conditions to favor spread. And there we can see in particular what's happening in Cameroon in terms of the hazard. And we're discussing possibilities of should Nigeria actually be thinking about supporting surveillance in neighboring countries so that they get an early warning of anything that's likely to affect Nigeria. So if I could see all the audience, I would now see, as I put up concluding remarks, a sense of relief perhaps in the audience. I'll just move this back so I can see you. So I often like to quote G.E. Box, who was a famous statistician, that all models are wrong, but some are useful. The wheat rusts continue to pose a major threat to wheat production. Modeling provides, I would argue, a valuable tool to prepare for and prevent epidemics. So the preparing for is the seasonal, pre-seasonal, working out what's the risk, what should we be doing, what varieties should we be growing. So we identify regions at risk from new races, early warning and advisory system within season. Then on cassava brown streak, and there are other examples that I could have given, it is a major threat to food security. Nigeria, 200 million people, more than 50% of that 200 million rely upon cassava as a staple. Excuse me, I'm just clicking. The virus has reached central DRC. And the models are enabling prediction of the arrival time, which I haven't shown you, but you can predict the distribution of the arrival time and the strategies for early detection and management. And thanks, I showed you the team and some of the key individuals as we moved through. It's really great to have an exciting team here and to work internationally. And also I'm really grateful. A lot of funding from the Gates Foundation, from the Foreign Commonwealth and Development Office under UK Aid, this ARC, and also from one of our research councils. Thank you. And thank you for listening, everybody.
Thank you very much, Chris. And I saw a question. I'm gonna let Brendan unmute himself, so I'm gonna ask him to unmute. So you can ask your question, Brendan.
Yeah, so my question was, well, at the beginning of your presentation, you showed these animations of spore dispersal and deposition. What did the data collection for those look like, and what was the extent of data collection for those to build those animations?
Let me separate that out. So the data collection, those are animations, and they're run from setting up a series of principles as to what we think is behaving. Then we take the computer output and we run it and produce those animations. Those are not based on any observational data, because you just can't get that amount of observation for the spores. So I wasn't sure whether, Brendan, you were asking, I'm just looking to see where, I'm looking at where it says Brendan now. Don't you wish that, do you not, oh, I'll answer the question, but do you not wish that Zoom would be systematic and show the same arrangement of people's faces and names for everybody? Then it, you know where you're looking. That's just a sideline of mine. So I wasn't sure whether you were asking particularly about the computational that went into that, or whether you were asking, did we have real data on where spores were?
Yeah, I was asking about if you had real data to kind of back those up.
The real data are, where is the infection occurring? Those are surveillance data. Those are real. They trigger where the spores come from. We predict where the spores are going to land, and then we compare where we say spores will land and where they won't land, and that's where we're doing the testing of the model. And I didn't go into that because it's quite complex, but we do have very encouraging data now for the comparison of the predictions with what we're picking up on surveillance.
Gotcha. That makes sense. Thank you.
But you need an awful lot of spore trapping to be able to do this. And historically, back in, excuse me, excuse me, a bit dry. Back in the 1930s and 40s, when even I wasn't around, they used to take planes up and actually take spore traps with them, and then look at long distance movement of spores. And I think we need to do that again.
Sounds like a lot of work, but very useful.
Yeah, indeed. Thanks, Brendan.
Thank you.
Okay, any other question? I have several questions. Let's see if somebody raise his or her hand. Okay, I don't see any.
Let people ask orally if they want without putting it in the chat.
Yes.
But no obligation.
Yes. So one question here. So thinking and see, this goes back to our previous conversation on the need of stochastic models and more powerful models that will tell a more complete story of what's happening, because reality is very complex, but at the same time we need to develop these models that that also go down to the simplicity of things. But now, so thinking about the future, because historically we know that we have depended upon meteorological models, mainly because we had the sensors, and so we can collect the data. But things are changing now. So theoretically, we can deploy other types of sensors and collect information. So now, what is your vision in terms of the next generation of models and incorporating maybe more powerful methods and systems that can tell us a much better story with the current sensors?
I think it's going to be really exciting. And I occasionally think, will we need models anymore? If we have enough data, and the sort of models I develop, if we have enough data, do we just need a simple machine learning and we've got it? I doubt it, but let's wait and see. So there are new sensors where I know there are groups working here in the UK, and I'm interacting with them, where they want to develop sensors that they'll know if a spore lands and they'll be able to identify that particular spore. Even so, and they're trying to make these smaller and smaller, you'd still need an awful lot of those to be able to get to use them realistically. And I'm all in favor of us trying this. Where I think we will be going is as follows. I hesitated because I now want to say two things. I think a lot of surveillance is badly carried out at present. I'm excluding Purdue from this, but people look in the wrong places at the wrong times. And then they say we don't have disease, and we don't have disease sometimes because they haven't looked when they needed to. I think the epidemiological approach can help with identifying where and when to look. And the same in working with, the same interacting with people developing sensors, where and when to look and at what density and how to make use of the information that comes out from those sensors in an epidemiological way, I think is going to be a very constructive way forward. I hope I'm not just making a case for the need for epidemiologists to continue.
I completely agree with you. So definitely looking at the right time, right place, as you know, even our group has demonstrated in some cases. So a follow-up question then, because you talked about risk, and so with this question, I'm trying to make a parallel with the current pandemic. The perception of risk. We have models, we have the capacity to develop those models and make decent predictions. However, we know that models in many cases are underutilized. And when it goes down to the decision maker, sometimes that information is not even used or is dismissed. So what do you think we should improve in the future?
I think that we need to work with policymakers, and that's something I've done in the UK. I chaired a UK task force on assessing risk and what we would do for plant disease in the UK. Quite some, I think it's about 5 or 6 years, probably a little bit longer than that. And that changed how our policymakers worked. What I also find very useful is are the following thoughts. One is that we produce user-friendly models. So that's with movies and sliders, and you can say, look, Minister, if you do this, let's just move these and say you carry out this sort of control, you put this amount of money into it. This is how good it could be, this is how bad it could be, which comes from the stochastic approach. Those I'm finding are successful. We worked with them through Tim Gottwald with citrus growers in Florida and elsewhere. We're using them in Africa with government ministers. Next point is to bear in mind that there's an asymmetry in the decision making that policy makers make, that politicians make over control. If they recommend doing something and disease is controlled, people are happy, but they're not necessarily aware that it's actually cause and effect. If the policymaker fails to do something and you get a devastating disease, nobody is happy. So there is an asymmetry in the different types of ways that you can be wrong and you can be right there. And I think we need to work with them in thinking how to handle that. Type 1 and type 2 errors.
Yes, very good. Thank you.
But having some friendly models, as I said to one group earlier on today, if I show a page of equations, people might be impressed, but they won't really be impressed. They will ignore it. But if I can show them a model in action and what the outcomes could be and the variety of outcomes, then they're more interested. And then they will do it.
Very good. Very good. Thank you. I see maybe Mariela has a question, or somebody else from my group, or somebody else from the audience. Okay.
It's getting close to your
Yes.
lunchtime, I imagine, isn't
Yes. And it's getting late for you too. So yes.
Yeah, I just wanted to say thank you to the professor for the seminar and for answering all the questions.
Good. You're very welcome.
Well, thank you very much again, Professor Gilligan. We are going to be in touch. I promise to email you back with some additional questions. And so hopefully we can continue the conversation.
Great.
Thanks again.
Thanks, everybody, and thanks for the opportunity to talk to you. And thanks to Cindy for the coordination. Much appreciated. Okay. Bye, everybody.
Bye-bye.
Thank you.
Bye.